Namaste all, I'm JVN Sunita Shekhawat, Faculty of Physiotherapy and Diagnostic, delivering this video lecture on 19 September 2023 on the topic coagulation of blood. Okay, so let's start with the topic. So basically, coagulation is the process in which the blood loses its fluidity and becomes a jelly-like mass few minutes just after it's shed off or collected in a container. Right, so basically when any kind of injury or collection of blood occurs, so after some time its fluidity changes into some jelly-like substance which is known as coagulation. Coagulation occurs because of some cascade reactions which occurs in different kind of factors. So basically we have 13 kind of factors which are responsible for the coagulation of blood. All these factors are activated in the form of one by one cascade manner. So clotting or coagulation mechanism is known as cascade theory. So according to this, we have all 13 factors on this slide. So here you can see factor number 1 is fibrinogen, factor number 2 prothrombin, then factor number 3 you have thromboplastin. After that, factor number 4 is calcium, factor number 5 labile factor, factor number 6 presence has not been proved. It means we have not discovered yet this factor number 6. After that, we have factor number 7 which is stable factor, factor number 8 anti-hemophilic factor, anti-hemophilic globin also, and factor number 9 Christmas factor, factor number 10 Stort-Fowers factor. After that, factor number 11 which is plasma thromboplastin antecedent. After that, factor number 12 which is Hegman factor, also known as contact factor. And the lastly, factor number 13 which is known as fibrin stabilizing factor or fibrinase. So all these factors help in the formation of coagulation, or you can say in the formation of clot after any kind of injury or collection of blood. So overall, this clotting mechanism occurs with the help of enzyme cascade theory. So basically, what is cascade? Cascade is the mechanism in which one step activated the next step, like these all 13 factors. So in starting, all these factors are present in inactivated form, which is known as proenzyme. So basically, proenzyme is be activated into enzyme to enforce the clot formation. Like you can say the proenzyme which is in activated form will be activated with the help of any kind of factor, and that result in the formation of clot. So basically, it is an proenzyme enzyme conversion reaction, which is a cascade type of reaction. Because of this cascade of reaction, at the end of this reaction, clotting of blood occurs with the help of a thread-like network, tightly formed thread-like network of the fibrin molecule. Right? So this enzyme cascade theory explains how various reactions involved in the conversion of proenzyme to active enzyme takes place in the form of a cascade manner. Cascade is basically refers to that the occurs through a series of steps. Each step initiates the next, and until the final step at the time of clot formation. Right? So now we have to see, or we will see about the clot formation mechanism, which occurs in 3 different steps, like you can see on the screen. So first step is the formation of prothrombin activator. After that, second step is the conversion of prothrombin into thrombin with the help of this above prothrombin activator. And the last third step is the conversion of fibrinogen into fibrin with the help of this thrombin. Right? So complete this clot formation occurs in 3 steps, and the first step is the formation of prothrombin activator. Second step, the conversion of prothrombin into thrombin, and lastly, the third step is the conversion of fibrinogen into fibrin. If we talk about the first stage, which is the formation of prothrombin activator, it occurs with the help of 2 different pathways. One is the intrinsic pathway, and the second was the extrinsic pathway. Both these pathways result into the formation of prothrombin activator, but with different methods, right? So as you can see, blood clotting commences with the formation of substances like prothrombin activator, which convert prothrombin into thrombin. It is formation is initiated with the substances produced either within the blood or outside the blood. The formation of prothrombin activators occurs in two different pathways. One is the intrinsic and the second one is the extrinsic. So let's move to the first mechanism, which is intrinsic pathway for the formation of prothrombin activator. As you can see in this diagram, it is the complete coagulation mechanism which shows all the three stages of the coagulation. The yellow part and light blue part. The yellow part indicates the intrinsic pathway of first stage, and while the light blue part indicate the extrinsic pathway of first stage. So basically first stage involve the conversion of the formation of prothrombin activator with the help of one is the intrinsic and the second was the extrinsic pathway. After this, the second stage is the formation of thrombin with the help of prothrombin. So prothrombin is converted into the thrombin with the help of the above factor, which is formed in the stage 1, which is prothrombin activator. And then lastly, this thrombin helps in the conversion of fibrinogen, which is present in inactivated form, and then it is converted into the fibrinogen, which is in activated form with the help of thrombin. And finally, this fibrin form a loose network, and then it form a tight network, which is the actual clot formation, right? So this is the basic framework of coagulation of blood, which occurs in all these three steps. First stage, prothrombin activator formation. Second step, thrombin conversion, and third step is the conversion of fibrinogen to fibrin, right? So we have to discuss all these factors individually. So let's move to the first pathway for the formation of prothrombin activator, which is known as intrinsic pathway. So as you can see in this diagram, intrinsic pathway start with the damage of endothelial cell and exposure of collagen. Because of some kind of injury, collagen exposed and endothelial cells damage, right? So both these factors activate some kind of factor which helps in the formation of prothrombin activator. So you can see in this diagram, let's see, first part is the during the injury blood vessels is ruptured. Because of this, endothelial cells are damaged and collagen beneath these cells is exposed. Because of this exposure, here factor number 12, which is Hagman's factor, comes in contact with the collagen and it is converted into the activated form of factor number 12, right, in the presence of callican and HMW kinogen. So first step is the because of the exposure of collagen, factor number 12 is converted into the activated form of the factor number 12 because of the presence of callican and HMW kinogen, right? After the conversion of this activated form of 12th factor, this factor activate factor number 11, right? So factor number 12 basically help in the activation of factor number 11, right, with the help of HMW kinogen. In the third step, you can see factor number 11 helps factor number 9 to be activated with the help of calcium. After that, factor number 9 with the help of factor number 8 and calcium activate the factor number 10, right? So this is the basic intrinsic pathway in which you can see first step is the activation of factor number 12. After that, activation of factor number 11. Next is the activation of factor number 9, and then lastly the activation of factor number 10. So this is the intrinsic pathway for the activation of prothrombin activator. At last, you can see factor number 10, which is activated because of factor number 9, involves with the factor number 5 and activate prothrombin activator. So as with this involvement, you can see because of this exposure of collagen, platelets also get activated and which secrete phospholipid. Phospholipid basically help factor number 5 for the formation of, or you can say activation of prothrombin activator, right? So factors activation is the one method. Second method which involved with this is the platelet activation and release of phospholipid. Both these factors, like for one is the activation of factor number 12, second is the activation or release of phospholipids, plus factor number 5, all these involved in the formation of prothrombin activator. So this is the intrinsic pathway. So now let's move to the extrinsic pathway. In case of extrinsic pathway, as you can see in this diagram, extrinsic pathway involves the formation of prothrombin activator is initiated by the tissue thromboplastin, right? You can see in this diagram. So tissue thromboplastin, which is formed on the injured tissue. So you can see because of the injury or damage, the tissue thromboplastin get initiated. This tissue thromboplastin contain proteins, phospholipids, glycoproteins, which act as a proteolytic enzyme. So basically these glycoproteins, phospholipids, involved with the factor number 7 and activate factor number 10. So there are three factors. One is the glycoprotein, second is the phospholipids, and along with factor number 7, these all the three components activate the factor number 10. After that, factor number 10, calcium, and factor number 5, all these components help in the activation of prothrombin activator with extrinsic pathway. So from this, prothrombin activator is activated from extrinsic pathway. So let's summarize this. Because of the damage, thromboplastin get initiated, which secrete glycoprotein, phospholipids, and these components along with the factor number 7 activate the factor number 10. After that, factor number 10 along with the calcium and factor number 5 activate the prothrombin activator. So here is some of the first stage, which is the activation of prothrombin activator with the help of intrinsic as well as extrinsic pathway. At the end of both these pathway, factor number 10 is activated, which result in the formation of, or you can say activation of, prothrombin activator. After that, we move to the stage number 2, which is the conversion of prothrombin into thrombin with the help of prothrombin activator, which is formed in the stage 1, right? So let's move to the stage 2, which is the conversion of prothrombin into thrombin. So blood clotting is all about the thrombin formation. Once thrombin is formed, it will definitely lead to the clot formation. So for the formation of thrombin, let's see some sequences. So first stage is the prothrombin activator that is formed in the intrinsic as well as extrinsic pathway convert prothrombin into thrombin in the presence of calcium, right? After the formation of thrombin, once formed, initiate the formation of more thrombin molecules. The initially formed thrombin activate factor number 5. Factor number 5 in turn accelerate the formation of both the extrinsic as well as intrinsic prothrombin activator, and which convert the prothrombin into thrombin. So basically this is a kind of positive feedback control. So let's see in this diagram. Here is the prothrombin activator. This helps in the conversion of prothrombin into thrombin. This is the sequence number 1. After that, this thrombin helps in the formation of more prothrombin activator, both in intrinsic as well as extrinsic pathway. So as you can see in this diagram, here comes with the positive feedback in intrinsic pathway, and here comes with the positive feedback in extrinsic pathway. So basically thrombin plus factor number 5, both these help in the formation of prothrombin activator, and finally prothrombin activator convert more prothrombin into thrombin molecules. So this is the kind of positive feedback which is controlled with the help of thrombin. So now let's move to the third stage, which is the final stage for the formation of clot formation. In case of third stage, the conversion of fibrinogen into fibrin with the help of thrombin, right? The final stage of blood clotting involves the conversion of fibrinogen into fibrin by thrombin. So you can see in this diagram, thrombin convert, or you can say activate, fibrinogen into activated form by removal of two pairs of polypeptide chains. So here comes is the inactivated fibrinogen, while here comes with the activated fibrinogen. So difference is the removal of two polypeptide chains. So now this activated fibrinogen is converted into fibrin monomer. After that, this monomer is get polymerized and form a loose strand of fibrin. This loose strand of fibrin along with the factor number 13 and calcium form a tight blood clot, or you can say tight strand of fibrin, which is help in the help in the trapping of dead RBC, WBC, which are present here for the removal of other infectious material. So this tight clot along with this dead RBC, WBC form a clot, you can say a network of fibrin, RBC and WBC, and this is the final result of blood clot. Okay, so later these loose strands are modified into dense and tight fibrin thread by fibrin stabilizing factors, which is factor number 13, in the presence of calcium ion, and all the tight fibrin threads are aggregated to form a mesh network of stable clot. So these three steps involve the formation of blood clot. So let's summarize all the three steps. So in first step, you can see this is the first step, result in the formation of prothrombin activator with the help of two pathways. One is intrinsic and the second one is extrinsic. Both these pathways and result will be activation of factor number 10, which involved with the factor number 5 and calcium, and activate with the activate the prothrombin activator. After that, this prothrombin activator activate the prothrombin into thrombin, while thrombin also initiate its own production with the help of positive feedback. After that, in next stage, this thrombin help in the activation of fibrinogen into activated form, which is fibrin. Then this fibrin monomer get polymerized and form a loose strand of fibrin, and finally with the help of factor number 13 and calcium, this loose strand converted into a mesh type of network, or you can say tight blood clot, and this is the stable clot. So here we complete with the topic, which is coagulation of blood, and as you can see, these are with the help of 13 factors which are discussed in the first slide. So basically, what is coagulation? It is the clotting of blood, or you can say conversion of fluidity into a jelly-like material. It occurs with the help of 13 factors, or the complete mechanism occurs in 3 stages. First step is the activation of prothrombin activator, second is the conversion of prothrombin into thrombin, and the last is the conversion of fibrinogen into fibrin, right? So thank you so much for this lecture.